Good afternoon, everyone, and welcome to our third seminar in the series of Ancient DNA. So we started off with a seminar about 4 weeks ago now by Hernan Barbuno on ancient crop DNA. And then last week we met Ludovic Orlando, who spoke about horse domestication. And today we're back in our comfort zone again with plants, and we've got a virtual visit by Nathan Wales. So just a few words about Nathan Wales. Well, he's not from Wales or even England or Scotland. He's from the United States of America, and that's where he did his bachelor's. Graduating first with a Bachelor of Science from Truman State University in 2005, and then an MA from the University of Connecticut in 2008. And then he stayed there and he graduated with a PhD in anthropology in 2012. During his PhD, Nathan, he also spent formative years in the Center for Geogenetics in Copenhagen, and his love affair with Copenhagen continued with a 4-year postdoc from 2013 to 2016. And this was followed by a postdoc in the University of California, just a short one for 1 year, a very short stint in France, and then he moved to his present position in the University of York, where he's a lecturer. So during Nathan's scientific journey, he's come to specialize on the recovery of ancient DNA from botanical specimens and using these to study crop domestication. But his interests are wide-ranging and also include such diverse topics as Neanderthal clothing and the evolution of wolves, and the evolution and history of grapevine cultivation in ancient Georgia. But without further ado, I'll hand over the screen to Nathan, and we very much look forward to your seminar. Thank you.
All right, well, thank you very much for that lovely introduction, and it's a pleasure to talk to all of you today. I'm going to start sharing my screen, so let's see if this works like always. All right. And let's see, hopefully, does that work okay for you all?
Yep,
right,
that's
great.
perfect. Thanks a lot.
And of course, if anyone has questions, feel free to drop them into the chat window. I might not see it right away, but I'll get to it eventually. Let me just kind of minimize this little screen for myself. All right, so as you just heard, I'm a lecturer at the University of York in York, England, and I'm a part of what's known as the BioArc facility. And what that basically is a combination of researchers doing work on archaeological material from a number of different time periods using DNA, but also proteins and other methods. So quite diverse. And what I want to tell you today is how I'm using ancient DNA to understand the history of crops. And I'm going to have, oops, there we go, just to give you a little idea of what this will cover. So I'm going to start by talking about domestication of plants in general. And I realize that probably many of you are already working on these sort of topics, so it'll be familiar. And then I'm going to tell you a little bit about how I use ancient DNA and some of the challenges that we face. And you've heard some of these things, I think, the last couple of weeks from Hernan and Ludovic. So I'll kind of specify a bit more on plants, move through that maybe a bit more quickly, and then tell you about some of the case studies of ancient DNA work that I've been doing on archaeological plant material, specifically focused on maize, sunflower, and grapevine. One thing I like to start off with when talking with domestication and showing archaeologists and archaeology students, the thing that usually comes to mind when we talk about domestication are animals. And this is a big area of research of ancient DNA, but also of archaeology. So as you heard last week with Ludovic's talk, the history of horses is very important, but so are these other creatures. And to keep it very simple, what I mean when I'm talking about domestication is that this is the process in which humans are controlling the reproductive strategies of an organism and selecting for desirable traits. And even though we often focus on animals, there are a lot of other things that are domesticated. So fungi, we can think about Darwin's famous pigeons, but of course we also have plants, which provide all sorts of nutrition. Usually when I present to a room of students, this is the face that I get back when I talk about my interest in plants, and people wonder, why exactly should we care about plants in the archaeological record? And I just want to give you a few quick reasons why I think it is important to look at plants. First, these domesticated crops allow societies to grow very large. So crops are the foundation for horticulturalists, for civilizations, and for empires. Now that's true if we think about the Fertile Crescent, things like wheat, barley, cereals, but it's also true in areas like Mesoamerica, where maize is what allows these societies to grow so large. And it's through these consistent yields of things like wheat and maize that populations can increase, populations densities can increase, and that ultimately leads to things like urban development, social stratification, and all the other great things that come with it. Another important reason, I think, to investigate plants in the archaeological record is that they are really a part of the cultural traditions in many different societies. So if you look around the world and you think about what plants are incorporated into the foods, whether that's in like Mexico or India, Italy, Japan, or something more local to the UK, these things become a kind of iconic part of a culture. And in each case, we have plants being represented. So it's important to understand where those plants come from, how they evolved with the cultures that become a part of them. And the last kind of perspective on plant domestication is the incredible genetic and phenotypic changes that happen during this process. And there's this, of course, classic example of brassica, where depending on how people were selecting upon this one species, many different exciting foods come out of it. So all of these come from this single species, and by understanding these genetic changes, potentially we can have a better understanding of how things happened in the past and potentially where things are going in the future. So that's why I study plant domestication. It is my core interest, and there are a few terminologies that I think are worth exploring just to make sure everyone is on the same page, and probably most of this is familiar to you all. The first is this domestication syndrome. So these are the changes that tend to happen in plants through the process of domestication. So things like larger fruits and seeds, loss of dispersal mechanisms, a decrease in bittery substances, loss of seed dormancy, and synchronized flowering. So most of those things are bad for a plant if it was to be on its own in the wild. But for cultivation purposes, these things are quite desirable to humans. So it's exciting to figure out how this happened. But the question as an archaeologist is, how can we actually see this happening over time? The way that people have traditionally approached this in the archaeological record is to look for small pieces of evidence that provide indications that there was domestication going on. And a very good example of this comes from wheat, where we have an understanding of what wild wheat is like. You can see here we have a wild emmer that is going to just naturally detach the seeds when they're ripe and ready to get spread. That is not a good strategy if you're a human who wants to collect these seeds. So instead, we have these non-brittle rachis, which keep the seeds attached. And if you look in the archaeological record, you can look for the specific scars that are associated with these two different types of wheat. And it's through this sort of evidence that people have had indications of when and where plants were domesticated. So this is using just conventional archaeobotanical approaches, and it has gotten us quite far to understanding where domestication took place. There are multiple different regions around the world where people now recognize plant domestication probably took place. The earliest seems to be in the Fertile Crescent, where wheat and barley were domesticated somewhere around 10,000 years ago. Other early evidence for domestication comes from Mesoamerica, where we have maize and squash domesticated probably slightly afterwards. and rice in East Asia sometime around 8,000 years ago. But you'll see there are many other areas of the world where plants appear to be independently domesticated. And these are areas where there's a lot of potential for future research. So despite that knowledge that we do have from conventional archaeobotany, there's a lot of big questions that still remain about plant domestication. First, when were these plants domesticated? We have some evidence already, but we don't always understand the sequence of these speciation domestications. Also, the pace of selection is unclear. Was domestication very quick, or did it take a long time where there was selection on different genes over thousands of years? There are questions about where domestication took place, whether it was a single domestication event or if there were multiple. And if we think about it from the human side, why did this happen? Did people intentionally start domesticating species once they knew that it could be done? Or was this completely unconscious? And if we think about it more as a geneticist, how did this actually happen? Was there certain genes that were selected very strongly, under very strong selection? Were there just a few genes that were under selection, or were there many small genes that had incremental effects, and that led to what we have today? So there's all of these questions. And the way that I am approaching these questions in plant domestication is using ancient DNA. And this is how I see ancient DNA from archaeological plant materials fitting into this picture, how we can answer some of those big questions. In the wild, we of course have a certain progenitor species that may look almost nothing like what we have today with the modern varieties. And archaeologically, there are going to be some early domesticates. So this is what it might look like phenotypically. If we look at it or conceive of it as kind of the genetic perspective, there's a lot of genetic diversity in the wild species. And that's represented here by all these different colored balls going through this funnel. And through the process of domestication, some of these alleles get lost over time. And so the modern varieties that we have are relatively inbred compared to the wild progenitor. And what I find quite helpful is to use ancient DNA in this intermediate step to understand what happened, what colored balls, if you want to think of it that way, what was happening, what was lost between here and what changed afterwards. And that gives us insights into the genes that were really interesting to people early on and what has been a more recent contribution, which may have impacts for things like breeding purposes. It's very likely that you saw this similar image in one of the past talks last couple of weeks, but it is kind of hard not to talk about Jurassic Park when talking about ancient DNA. And it actually is kind of helpful to think about it because it has a lot of, it has had its impact on the field in a way. The reason is that during the 1990s in particular, a number of researchers were trying to get DNA out of ancient dinosaur fossils. For example, there are these two papers from 1995 where they tried to amplify DNA from this Cretaceous dinosaur egg. And lo and behold, they were able to do so. And they published it in a very high profile journal. And you can still find this ancient dinosaur DNA on GenBank today. And if you test it and you do a blast analysis of this putative dinosaur DNA, what do we find? It matches almost perfectly with E. coli. And this was the time in which ancient DNA came to realize some of the limitations of the field. At this point, there weren't very good reference databases to understand what DNA was actually recovered. So this was E. coli. And contamination, it was then recognized, is a major problem. And we also did not fully understand the process of DNA degradation. We now think that dinosaur DNA cannot preserve because it is simply too old, but samples which may be up to 1 million years old might contain DNA, as some woolly mammoth DNA has been recovered. So based on those discoveries, a lot has been done to figure out what happens to DNA over time. And we now know that once an organism dies, the mechanisms that are there to repair the DNA, they stop working. So DNA will continue to break down more and more over time. The DNA molecule is attractive to microorganisms. It's full of sugar and phosphate. So microorganisms will come, eat the DNA, meaning that there's less of it over time. And then finally, this figure shows the DNA length recovered from a 3,000-year-old sunflower. What we see is that the DNA is super short after even a couple hundred years. So instead of DNA being millions of base pairs in length, it's only a couple tens or maybe 100 base pairs. In addition to this fragmentation, it also becomes chemically damaged. And you may have heard about that in the last couple of weeks. And I'll show you some examples of it. But one of the good things about this DNA degradation is that we can sometimes use it to authenticate that the DNA from a given sample actually belongs to that sample. And what that means is that I can take an archaeological sunflower, I can look at this fragmentation pattern, I can look at the damage pattern, and I can check to see if it makes sense. And sometimes this actually has been quite helpful to recognize an intrusive sample, where once we did radiocarbon dating of it, we realized actually it wasn't as old as we thought it was. So this is quite important for using ancient DNA research. Now, based on those challenges and recognitions that DNA is easily contaminated, labs have been developed, like the one that I have here at York. And this is a part of what's known as BioArc, where we're a collaboration between biology, archaeology, and chemistry. And we go to great lengths to make sure that we don't contaminate the samples that we have. So we operate in full body suits, we use a lot of controls, and when we get DNA from an archaeological sample, then we can be quite confident that it actually belongs to that sample. In terms of the methods that I use, I think you heard a lot more from Ludovic and Hernan, but just to give a simple summary, for the most part, I'm using high throughput shotgun sequencing. And with that, I take an archaeological sample like this maize cob or proto-maize cob that's something about 5,000 years old. I do an extraction of that. I get out a whole mixture of DNA. It's not just DNA from the plant. It's going to be bacteria. It's going to be some fungal DNA. There's going to be human contamination. And we sequence all of it. You get a lot of data in the end. And then generally what happens next is you map it to a reference genome. Sometimes we do a blast analysis, but we then can infer what DNA is a part of the sample. And I hope in most cases, these archaeological samples, if it's a maize sample, I hope it has at least, let's say 1% maize DNA. So in reality, there's a lot of bacterial DNA that gets recovered. And sometimes we then have to turn toward things like enrichment, where we target SNPs of interest and we use in solutions enrichment to pull them out. So a lot of exciting things that can be done in ancient DNA on plants, and I'll show you some case studies in a moment. But one of the big things to remember is that even though there are a lot of archaeological plant remains, most of them are not suitable for DNA. And the reason is that most of these plant samples get preserved by charring, where they end up looking something like this. They get heated to a high temperature. That preserves kind of the structure of the seeds or tissues, but it also destroys the DNA molecules. And so some colleagues and I tested a number of charred plant remains, and we showed quite convincingly that they almost never have anything in them that can be useful. On the other hand, what does work are desiccated plant remains and waterlogged plant remains. So these are limited to certain environment types like dry caves or waterlogged areas, latrines. But in those cases, we can get very good DNA preservation. And that's where the samples that I'm going to tell you about today come from. And before I jump into my case studies that I'm very proud of, I want to point out that there has been a fair amount of DNA work on archaeological plant remains on different species. So some of this kind of goes under the radar a little bit compared to the big studies on ancient human DNA. But you'll notice all of these different species on the left have been tested using high throughput sequencing. So these are up-to-date modern methods, and they come from different time periods, mostly within the last 5,000 years or so. And some of the early questions about domestication are still possible if we think about these early steps. If we find the right archaeological sample, these type of things could be investigated in maize barley, and other species. All right, so let me tell you about my first case study. This is a collaboration that I've had for quite a while since my time in Copenhagen, and it continues to grow in new ways, and that is on maize. So maize was domesticated in Mesoamerica, in what is today Mexico. And based on modern DNA, the genomic diversity of the Parviglumis, it's quite clear that it comes from the Balsas Valley of Mexico, so here on kind of the west or southwest coast. And some of the earliest archaeological samples come from the highlands, like Tehuacan Valley. And so you'll see that there are these different climatic regimes, and there's been questions about how exactly maize was domesticated. If it did happen here, what was some of the earliest steps, what genes were under selection. And so we've used archaeological samples, especially from the Tehuacan Valley, but also from the United States, to think about this process of domestication and improvement. In my opinion, this is one of the most staggering examples of what happens during domestication. So on the left here, we have Teosinte. This is the wild progenitor. And you can see the plant has this very branching structure. And what is analogous to the cob is a little tiny tissue. You can see it here with these enclosed capsules. And what becomes the maize cob that we know, it's much larger. The plant itself becomes just not this branching structure, but a single stalk. So there's a lot of things that have happened in the process of domestication of maize. And on top of that, compared to what's in the wild with these small Teosinte cobs, if we want to call them that, modern maize has a huge amount of phenotypic diversity. So how exactly this has happened? I mean, it's happened obviously within the last 10,000 years, but this is astonishing. It means that there has been selection, and in individual areas, different things have been important to people. So it's interesting to try to figure out how this actually occurred. So my colleagues and I have tested a range of different maize samples. The first one I wanted to tell you about is kind of more about the early part of this maize domestication history, where we had access to this 5,300-year-old cob from the Tehuacan Valley of Mexico. And note, this is still kind of halfway back in the domestication of maize, but it is one of the oldest available tissues that is not charred. We recovered a huge number of SNPs, and we enriched genes of interest. And we could find, using a genetic analysis, comparing wild Teosinte, so that's here in yellow and in purple. So this is the wild Parviglumis from the area where we think maize was domesticated. We can see this ancient sample is falling ancestral to all of the different modern land races of maize. So it doesn't mean that it was the direct ancestor to everything, but it was a part of this population. So the maize that made its way to the Tehuacan Valley does seem to be very closely related to the maize that then gets dispersed throughout the Americas and then eventually around the world. And we could also see some specific genes, trying to understand what had been under selection at this point in time. We can see that the gene related to the ear shattering and also genes in the starch pathways, they were still in the ancestral state. So some things had been clearly happening with domestication. So this looks different from Teosinte, but not all of the genes had been domesticated just yet. So this does indicate kind of a potential protracted domestication process where things happen at different times. Moving on from that one, another quick case study I can tell you about on maize is trying to understand how it is that maize left Mexico to go to other places. And to understand also what happens once it gets to those other places. And to do that, we tested a set of samples from the Tularosa cave in New Mexico. So this is in the United States. We were able to investigate two different hypothesized pathways into this area, either kind of along the coast or through the mountainous areas. And this was done using genetic distances between modern land races that are found in these areas. And it was clear that this pathway through the mountains had the stronger initial stronger evidence that these are descended from samples that came through that highland area. So that was exciting. But on top of that, we were able to look at 2 different time points within Tularosa Cave. We had samples that were about 2,000 years old and samples that were about 750 years old. We could tell just by looking at them that they look different. So this one is kind of small. This one is more of like a cigar shape. And we were really curious to know what genes were under selection in this 1,000-year period in New Mexico. And what we found is that the genes with the strongest evidence of selection were related to dehydration response. And that makes a lot of sense for a plant that has left the tropics of Mexico to go into this very arid climate in New Mexico. So we were able to look at this gene and we could see that there was reduced diversity compared to the wild, and it happened within this time window. So that does tell us something about the adaptation of that crop to that specific environment. And the last kind of intriguing part of the Tularosa samples is that we found 2 genes that were also under domestication or had been changing during that 1,000-year window, the AE1 and the SU1 genes, which are associated with sugar content and also pasting properties. So there are changes going on at this time. And in conjunction with what we know from the archaeological record, we can start to suspect that this is where people were using the plants in new ways. So this allows for the creation of things like tortillas. And it's plausible that this genetic evidence lines up with the archaeological evidence that people are using maize in a different way, starting sometime after 2,000 years ago. And I thought it would be nice to show you just some of the ongoing work. So this is unpublished. We hope to get it published soon, and it's still we're still doing some final analyses. But one thing that I've been quite interested in is understanding how maize reached Eastern North America. So the maize that was used by Native Americans in this area, in the eastern part of the United States, ultimately that maize is some of the maize that becomes really important commercially today. So that's maize that then gets exported around the world. And we've wanted to understand how that exactly happened. And to do that, we had access to a unique set of samples from what's known as the Ozarks in the Arkansas region of the United States. There are some really well-preserved maize cobs in these sites, in addition to the maize cobs that are preserved in the Southwest United States. We tested a huge number of samples, and we tried to investigate which way we thought these Ozark samples, how they came from other parts of the United States. Based on the genetic distances, we can see that a path coming up through this way is most likely. So there's not much genetic link between samples from Mexico and Texas, but rather there's strong links from this western part of the United States. And we can see that these Ozark samples are furthermore very closely related to the Northern Flint land races from Native Americans, which were grown in this part of the United States. So here's an example of a Northern Flint, modern Northern Flint maize. And these archaeological samples from the Ozarks match it very closely, which suggests that they're probably a part of the same population. And what's kind of cool is that, again, as I told you, this maize in the middle is the one that becomes really important commercially today. And it was formed in the 17th or 18th century when farmers crossed Northern Flint land races with Southern Dent land races, and you get something like this. So we can now have a little bit better understanding of where this commercially important maize came from. And hopefully one day that might lead to further information about breeding purposes. If we know the full history of these maize lineages, we may be able to better plan breeding purposes. The last slide, which kind of popped up, still related to this, we've also looked in this data set for evidence of selection, just like we've done for the Tularosa samples. And the one that's kind of an outlier, as we see in many different analyses with genetic diversity and this population branching statistic, the gene that looks weird is the WAXY1 gene. It has really low diversity in the Ozark samples. It seems to be the same allele that's in the northern flints. And this seems to be then something that was selected after maize left the Southwest. So it's specific to the Ozarks and Eastern North America. And we're still trying to understand what exactly that means, but it may have to do further with the use of the plant or maybe irrigation or something like that. So that's something we want to investigate in the future. Let me just make sure time-wise I'm all right. Yeah, so the other case study I wanted to do quickly is on sunflower. So this has been a project in collaboration with Ben Blackman at the University of California at Berkeley. And here the research questions are pretty similar. We're trying to understand when and where sunflower was domesticated, what genes have been under selection. And to do that, we again have access to some of the same archaeological sites that I just mentioned, the Ozark sites from Arkansas. And you can see there's some just incredible preservation of this disc and also these achenes, where these are not charred. These have really excellent DNA preservation in many of them. To show you the obligatory what happens in domestication with sunflower, the wild version of sunflower has a branching structure, just like with maize, many small heads, very small seeds, relatively low oil content. The cultivars that are produced today usually have just a single head, very large seeds, and high oil content. And land races, which were grown by Native Americans, some of them look like these cultivars, but some of them do have multiple heads. And so there's questions about how exactly these changes, when they happened. And so what we're doing with this is combining ancient DNA research along with some archaeobotanical and radiocarbon dating to get a good understanding of how everything has happened. A quick picture of when and where this occurred. We think that domestication occurred probably about 4,000 years ago. You can see in this little schematic. We know that Native Americans were collecting wild sunflowers before that, and then there is a change in the size of the seeds sometime around 4,000 years ago. And by 3,000 years ago, it's quite obvious that they are domesticated. There are questions about where exactly this has happened. There's an idea maybe there was an independent domestication in Mexico, although most of the evidence points to the Midwest or eastern part of North America. And there's some quite diverse uses of sunflower by Native American. It's been used, the oil as a food is for anointing hair. You can eat it, the achenes, like we do today, but one lesser known purpose is for creating dye. So there are a few lineages that have very purple dyes that are released. So this is probably a further improvement that happens much after the domestication. The challenge with looking at sunflower just with modern samples is that it has a complicated history in the last couple hundred years. So for the most part, it was ignored by colonists. It was taken to Europe. And in the last couple hundred years, there was very intensive breeding happening in Russia. And those cultivars then get reintroduced to the Americas. So if we were to look at the land races or the cultivars that are available today in the Americas, they may not be representative of what was there 400 years ago. So it's quite important to look at these archaeological samples. And so I've tested in the lab many different samples, but for the most part, as I told you, samples from Arkansas, some from Kentucky. These provide dried samples that do release DNA and can be tested. And we've so far focused on one publication, but we have some other data in the pipeline, some genomic data. But just to give you a quick synopsis of what we've done so far, we focused on the chloroplast or plastome genome to understand what diversity looks like in the wild and what it looks like in domesticated sunflower. So this is a haplotype network, simply all of the different haplotypes that are available in the chloroplast of sunflower. And these little dashes represent how different they are from one another. And to just kind of walk you through this, in the wild, there's a huge amount of genetic diversity, as we might expect for a wild species. So all of these green circles represent a unique sequence observed in a wild sample. And what we found is that the archaeological samples have the same sequences, which is expected, but it's a very small subset of what could have been taken from the wild into the domesticated group. So those are the ones in yellow. And what's interesting is that when we go from the archaeological samples to the land races, which had been grown by Native Americans, they have even less diversity. So this suggests kind of a continuous bottleneck happening through the process of domestication, where actually there may have been more diversity a couple thousand years ago than was present 400 years ago, let's say. And we can specifically see that certain lineages of these archaeological samples just disappear. Those did not make it into the domesticated population that we know of. And cultivars show evidence of introgression. So this was another important thing. We knew that sunflowers have been intentionally crossed with other sunflower species, like Helianthus pedicularis. And we can see that in the plastome sequence. So that is also kind of helpful for understanding how domestication and then recent breeding impacts things. And this also helped us identify some species that we thought were just pure land races. Those actually have some clear introgression from other species of sunflower. And that's really essential to take into account when we are going to do this whole genome analysis, where we try to understand what genes were under selection at different points in time. So based on this case study, what we can see is that indeed sunflower does seem to be domesticated at least 3,000 years ago. All of the chloroplast sequences that we have fit very clearly with modern domesticates. And there's a huge potential going forward to understand gene by gene what has happened. And I do think in the next year or two, we'll hopefully be able to release some of the publications about that. And not to overwhelm you with case studies, but I wanted to finish up on this project on grapes. And the reason that I wanted to talk about this is that it is conceptually really different. So the last species, the maize and the sunflower, they're annual species. But with grapevine, everything we have to think about it all totally new. So as you know, grapevine has a lot of different uses. You can use it especially in wine, and that's where most of it is used commercially, but it's used for many other products. And of course, also eaten as a table grape. In terms of what happens in domestication, there's a huge change in the structure of the berries, the size of the berries, the seed shape changes a little bit. But actually the most important thing with domestication is that it becomes hermaphroditic. So in the wild, plants are either male or female. The domesticated grapevine, almost all of them are hermaphroditic, and this is good because it means that every plant is going to produce berries, and you don't have to have some male plants to pollinate the others. And I've been curious about domestication, so we're shifting now into a whole nother part of the world. The history of grapevine is particularly important around the Mediterranean basin, and I've been working the last couple of years on a project with this. We know that probably grapes were domesticated in the Caucasus or maybe Turkey, perhaps around 8,000 years ago. It's a little bit more uncertain than in the other species, but we do have evidence from Georgia where it seems that people were collecting grapes and making probably wine around 6,000 BC. We find further evidence of wineries in Armenia, like this from Areni, about 4,000 BC. And we can see that then this grape cultivation and winemaking culture spreads across the Mediterranean, taken, for example, by the Phoenicians. And we find then winemaking facilities with the Romans and other groups for the last couple thousand years. And so I've been curious to understand how this process happened, and especially to understand the history of varieties. So if you're a wine connoisseur, you'll know that there are these very famous varieties like Pinot Noir. We want to know when those appeared and how they got established. And if we think about things like Pinot Noir, the thing that makes them so different than when we're talking about land races of maize and sunflowers is that these are genetic clones. You make Pinot Noir, and it doesn't matter whether you have it from France or you have it from Australia, the plant itself is the same Pinot Noir. It should be the same genome. And the same is true for table grapes. So these Thompson seedless grapes, wherever you grow them around the world, genetically, they should be the same. And this is all possible due to clonal propagation. So as you know, you can take cuttings of a grapevine, you can plant them, or you can graft them onto other rootstock. And you can do this again and again and again for centuries. And what it means is that these varieties get kind of frozen in time. And so this is how I think about my work on ancient grapevine. I'm trying to understand how it is that these varieties get established and how old some of them actually might be. So to give you just a last example of what comes of this, this is a publication from a couple of years ago where we did this application to a collection of grapes from France. So these are well-preserved archaeological samples that were provided by collaborators from France. In this case, they were all waterlogged and they date from the late Iron Age, a lot from the Roman period and some medieval samples. And I use the standard approach, going through all the steps, shotgun sequencing, but also importantly, the enrichment of 10,000 SNPs that we know are informative to differentiate between varieties. We went through our standard checks to see, did we actually recover ancient DNA? Yes, indeed, the DNA was short. It had chemical damage that I would expect. I was pleased to see that we had almost 9% grape DNA in the end, and we were able to get most of these SNPs that we were targeting. So really high success. So the DNA works. I just thought I'd show you on this one, we did kind of the same test for the sunflower and maize, but this is kind of the standard protocol. In terms of what then we can actually interpret biologically and archaeologically, here we have an MDS, or multiple dimension scaling plot of what how these archaeological samples fit into the bigger picture of our reference panel. The archaeological samples are in black. And in this first one that I'm going to show you, I'll show another one in a second. The main thing to notice is that none of them, or maybe one, fall within this kind of green area here. And those are the samples that are wild grapevine. All of these archaeological samples from France are falling within the domesticated compartment, which means that people probably were actively collecting these and managing these vines. If we redo that analysis without those wild individuals and make this plot again, we can see that most of these archaeological samples fit very closely with this purple group, which are today the vines that are used in winemaking in West and Central Europe. So this provides even further evidence that these archaeological samples, for the most part, were probably being used in winemaking. And that also fits more or less with the archaeological sites that they're found with, where they're often near to winemaking establishments, and it matches what we know about the Romans and other periods where people were actively producing wine. The other kind of cool things that we were able to do in this data set was to check to see if we could find matches to this reference panel and also between the archaeological samples. So this first one is that we were able to find some examples of archaeological samples that were genetically identical to each other for these SNPs. So that includes like these little blocks here. What this is a kinship coefficient showing that these samples are genetically identical to each other. For the most part, it was happening at the same site, but in a few cases, we were finding single individual seeds from two different places in France that were genetically identical. And so what this shows is that we do have clear evidence for the same varieties of grapes being grown at two different places in France during the Roman period, separated by 600 kilometers. So we know that the Romans had varieties, but here we have the first archaeological ancient DNA evidence that this was happening. And it provides a way for us to test this going forward. And for the last kind of piece of the puzzle, how is it that these archaeological samples match up with modern varieties? Well, we were able to do, again, with that kinship analysis, comparing this time not between archaeological samples, but to this reference panel. What this kind of messy network shows is that we do have archaeological samples here in orange from the Roman period that have a parent-offspring relationship with varieties now grown in the Alps. So taking into account the SNP diversity, the allele frequencies, we can see that these are separated by just one reproductive generation, even though it's 2,000 years old. We found even more of such relationships for medieval samples, and the one that was kind of most exciting of all, we had a perfect match from a sample from Orleans in France, dating about 900 years ago to a variety of grapes today grown in France known as Sauvignon Blanc. And we can now have direct evidence for this 9 centuries of propagation where the single variety was maintained in France, and it's called different things in different places, but it does have this really kind of special cultural contribution where it is seen as kind of a special thing within Eastern France, where it's celebrated along with cheese and nuts. And it does give us kind of that history of this cultural tradition now that we didn't have before. Despite all this exciting stuff, sometimes journalists can take this a little bit too far, and I had to kind of correct them. So some people might think we now know exactly what wine tastes like, and I think that's true to some extent. We know what some of the varieties that were grown. However, winemaking is a complicated process, and fermentation was spontaneous. So there's a lot of things that are involved in the final product of wine that we need to take into account before we could say things like this. So different yeast will change the flavor of wines. In the past, people were adding special things to the amphora in order to preserve it. For example, people often added pine resin, which gives it a very distinct flavor, but it also helps to protect the wine. So we can now have steps toward a better understanding of what wine or maize or sunflower were like in the past, but each time we need to think about what other factors might actually be impacting it as well. So to kind of step back and think about this in a bigger picture, I do see kind of a lot of exciting things coming out of ancient DNA with plants. It tells us a lot more about the agricultural practices in the distant past. It also can tell us about how people were using plants in the relatively recent past and how that may have changed quite quickly. We can use this in terms of agriculture to think about what species have been changing and how the genes have been impacted. And I think it also has a lot to do for consumers today who think about these things and eat all these products and better understand the long histories that many of them actually have. And thankfully, there's a huge amount of potential for future research. So there's a lot to do. As you can see in this map, many different species were domesticated around the world. And if we have a reference genome and if good samples are preserved in the archaeological record, we could potentially do this for dozens and dozens of different species. And I hope that is something that happens going forward. So this research was made possible by a huge number of collaborators and funders, and it would take forever to thank them all, but they are invaluable to me, and I thank each and every one of them. And I want to make sure I also thank you all for your great attention today. So thank you. And I'm, of course, happy to take any questions.
Thank you, Nathan. That was a very nice, very clear and didactic seminar. And I can see you're getting some a virtual applause.
Oh, thank you.
Okay, so typically for the discussion, we try and encourage the PhD students, postdocs, and other early career scientists to step in first with the first questions. Any PhD students, postdocs, others, questions for Nathan? You can use the chat box too, if you prefer. Yeah, Haneen.
Hello, thank you very much for your talk. It's more like maybe a technical or stupid question. So in the sunflower, when you did with the blastome, you were able to look at integration. So integration, but that is, I think, particularly integration happening through the plastids, but not actually nuclear, because that happens through pollens, right? So if it's like a nuclear, so this is somehow like really limiting you, so you're not getting the very full picture.
Yeah.
So what is your comment about it?
Yeah, good question, and no stupid questions. So that was great. Thank you. So it's a very good point. So we know that people had crossed other species of sunflower with Helianthus annuus intentionally to for male sterility. So historically, we know this has happened, and it is true that like the plastome is the part that counts, right? The chloroplast is what makes that happen. However, in order for the plant to be viable, probably there is still some of the genome of Pediolaris kicking around. So I don't think that they were able to fully back cross it out and only keep the plastome. So the way going forward, what we have to do is to look at some of those samples and try to understand maybe what genes are still present from Pediolaris in those land races that were crossed over. And I think that might actually be pretty difficult, because I don't think we have a good reference genome for Pediolaris. And so anytime when you're working on kind of like these species complex or closely related species, you have to have really good understanding of what the different ancestral genomes are. And I do think that that is going to be kind of a stumbling block for a while, maybe, until we generate a good reference genome from some of these other species. And we've had to do that to some extent, where we've been working on some samples from Mexico, and we don't have a good grasp of the diversity of sunflower Helianthus in that area. And so we've been doing some shotgun sequencing on things we think are other species or subspecies. And so there's actually a lot of legwork and money that has to go into like building up a reference panel before you can really use the ancient DNA in some cases. I hope I answered the question. I also talked all around.
Yeah, okay. Yes, thank you. I'm just actually, I just wanna, because I am looking at integration in wheat, but of course we have nuclear DNA data, SNP data. But then at some point I thought that I want to eliminate the integration, because integration also, like if I want to look for something such as, I don't know, diversity, then it is affected by integration. But if I want to look at, for example, variation that is not affected by gene flow, I thought like maybe looking at chloroplastic DNA, for example, but then still even the chloroplast move. So I think at some point you cannot really eliminate integrations.
Yeah, no, I think you're exactly right. And one thing which I didn't show, but we work on a lot. So we have SNP data from the modern samples. One thing we do in kind of the first step is we check just in a simple PCA, and we have the different species that are present, and then we check to see if any of our land races or cultivars start moving in the direction of some of these species that we think have been crossed with each other. And then that helps us to identify ones that are likely crossings or admixed individuals. And then we exclude them from the data set and redo the analysis and see how things look. So I think the plastome is one piece of evidence, but it could be that depending on the way things were crossed, you might not have the plastome get transferred, right?
Yes.
Yeah, it is very important to look at the nuclear SNPs for sure.
Yeah. Okay. Thank you very much.
Any other questions from the early career scientists? If not, I will ask a question.
Yes, please.
Okay, so it's building on from the last section of your talk, where you spoke about the domestication of grapevine, and the sort of intriguing question: what did wine taste like back then? And you spoke about the yeast. Can you not recover the yeast, maybe even in its live form, from some of these old vessels?
Yeah, so this is it's something that I've tried to work on, and I think it's also very controversial. So I've been skeptical of it. So there has been some publications where people have taken amphora and they have extracted DNA from it, and they've mostly been using PCR-based approaches where they amplified markers for like a tRNL barcode or RBCL barcode. And it has worked, but there's a lot of questions about whether it might have been contamination. Mm. With the yeast, there have been claims that people have resurrected yeast from a few vessels. So they've taken archaeological samples and they've been able to get yeast out of them. The question is that some sort of modern contaminate yeast, or is that an actual ancient one? And I don't think that people have really decided which one is right just yet. And I think it could be true. And I have some really intriguing pictures of like vessels with a lot of what look like yeast cells on them. And it seems like it's on the interior surface of the vessel, which would be consistent. But also sometimes when these studies get published, they seem to have very long DNA molecules, which doesn't match up with my expectations of DNA fragmentation. So I think that in a few years, we'll probably have a better picture of it. But if it does work out, that I think there is then potential to understand the taste of ancient wine or similar questions, because then you would have the full picture, the full microbiome of what was going on in that vessel, what species were present, and what flavors they might have added or contributed. Yeah.
Okay, and one more question. The seed that you I mean, these old archaeological seed, are any of those able to germinate? Because we've all heard of some of these ancient date palm seed that were found in Israel, and I think they were whatever 2,000 years old or so. So is there scope for anything like that, do you think, with these old vitreous?
I think that date palm in particular might be special, that it's really well designed to survive high temperatures and arid conditions. And I don't think that is true for maize or sunflower or grape. I've also been somewhat skeptical of those claims, although I have colleagues who are on some of the papers and I know they do good work. So I feel sort of conflicted, because I also would assume that just based on being in a hot climate for 2,000 years, fragmentation, I think, should happen. So I'm surprised that it doesn't happen more, and maybe only some of these seeds are able to germinate. But I've also been kind of worried about it, because if things germinate, then I'm sort of out of a job, right? Like, why do you need me to do ancient DNA if you can grow this plant yourself? So I think that it would be kind of a cool combined thing to bring together some of these germination studies as well as the ancient DNA and make sure that they lead to the same conclusion. But yeah, I haven't tried any of mine. Oh, I see there is one question from...
Yep, from Yong.
Thank you, Lesson, for your great talk. Can you hear me?
Yes, yeah. Thank you.
It's very nice stories from the ancient plants to the wild species or landrace to the current modern varieties. I was wondering when we saw the domesticated evidence from phenotype and provide evidence, we can identify these genes from the wild species to domesticated varieties. And the quality and the yield has been improved a lot. So I was thinking, and what do you value the possibilities, like from the ancient plants and to the current modern varieties? Do they still have a lot of potential to identify a lot of genes that have not been cloned or being filtered by chance? These genes, and is it possible to find more evidence to domesticate, to improve, or break through the bottleneck of the current modern breeding program?
Yes, very good question. So I think this is something that we've kind of promised and hoped in grant proposals, that if we understand the genes that are under domestication, that can give us some insights into some of the genes that may have been kind of hitchhiking, right? That were accidentally under selection and that have lost diversity, that maybe that's not ideal, right? So maybe some of the genes that are just upstream and downstream of the ones that were under selection, they might be quite useful to have diversity there for pathogen resistance or other things. And I do think that if we go back and we find, okay, this gene was under very early selection, it was very strong, it would make some sense to try to target the genes in the neighboring area and introduce diversity that maybe hasn't been introduced using conventional breeding approaches. So maybe with CRISPR or something else to say, let's add in some of that lost diversity and see what happens. I think that would be a really exciting way to kind of combine agronomic research with what's being done with ancient DNA.
Thank you.
Nathan, I have one more question. When did the hermaphrodite trait evolve? Because that must have really, almost overnight changed the complete sort of picture of diversity. It must be quite easy to pinpoint when that happened, right, from the resequencing.
So we know the genes that are involved in it. We don't have the right archaeological samples to answer it, unfortunately. So we think it happened pretty early on in the domestication history, but the archaeological samples that date to that time period so far haven't yielded DNA. So I don't actually know when it happened, but we know within the genus Vitis, most of those species are actually hermaphroditic. So it was a reversion to that. Mm-hmm. But it is kind of like the core question of understanding grape domestication. And I think it's just going to be a matter of finding the right sample to be able to answer it. And yeah, sadly, I don't know just yet.
Okay, thanks. Rod Wing.
Yeah, hello. It's a great talk. I'm sorry I'm talking on my phone right now. So
that's right.
Listen to the whole thing. The sunflower story, I had two questions with that. So I also had a question on why you chose the plastome over using just nuclear DNA. And have you done similar work with nuclear DNA in the sunflower analysis? And my third question with that is, in this cave in Arkansas, are there any other plant species in there that you're working on?
Yeah, good questions. Okay, so the plastome as an area of interest, in part that is because kind of historically ancient DNA has focused on mitochondrial DNA and other organellar DNA. And that's because there's many copies of it per genome or per cell. And then it's easier to do it with shotgun sequencing. So with a limited amount of shotgun data, you can have full coverage on the chloroplast genome for relatively inexpensive. We are doing nuclear DNA analysis on archaeological sunflower. We've generated the data, we've done some analyses, but we don't have the resolution yet that we have on maize. But we are planning to look at the genes that we know have been under selection and see what alleles are present in these archaeological samples, because that should tell us when things like flowering time and the size of the head, when that may have changed. So that is a really important thing. And for the sites that are in Arkansas, there's a big diversity of plant remains that have been recovered, and it's kind of published in obscure PhD dissertations from the 1980s, because nobody's touched it really since then. But I haven't started working on anything else, but I am curious about a few lost crops. So there's a species, little barley, that probably was domesticated and then has been lost. So it'd be interesting to know why it is that Native Americans originally domesticated a couple of these species and then why they gave up on them. And it might be because maize comes to the area and it's just such a great crop in comparison, but I think that is kind of a potential area for future research.
All right, thank you very much. Excellent presentation.
Thank you, Rod.
Thanks, Rod. And Nicola. Nicola, you mentioned you had a question.
Yes, sorry, I'm answering from the phone. And great talk. I might have missed one of the last parts of your talk about the grape trees. And my question is, so I know that these days most of the grape trees are actually grafting. So the root is taken from another plant and the stem is made from a different plant. And this is just to increase the yield. Can you actually tell by your analysis? And I know that in the grafting portion of the plant, there can be some shuffling, at least in that part, there can be some shuffling of cells. So there is some cells from the upper part that goes into the lower and cells from the lower part that goes into the upper. Can you see this thing by examining the DNA of the plant, if there is some heterogeneity, or is that like, that doesn't make any sense, because probably this is just a recent thing. Back in the days, they used to grow from a single plant, like they didn't used to grow.
Yeah, so I didn't get much into the grafting question, but it is a good point. So the reference database that we have, or that I use, comes from mostly French colleagues, but also some Italian colleagues. And I think that the samples that they're testing, I think it's always leaf tissue, where there isn't much risk of cells coming from the grafted rootstock. And in all of our analysis, we also include rootstock as one of like the populations. At least we check to make sure that we don't have rootstock, and we shouldn't find it archaeologically, because the movement of rootstock, so it's all American species, American Vitis species that were taken to Europe and elsewhere. So that happened within, last couple centuries. Archaeological samples should be much older than any of that introduction. So long as we don't find any rootstock, I think we should be all right archaeologically.
Okay. Thank you very much. It was just like a curiosity.
Yeah. Thank you.
Okay.
Any more questions? Oh, there's one in the chat box. Saula, do you want to join us and ask us the question?
Hello, thank you very much for the talk, Professor. I was just curious, since you mentioned the selection for the dehydration response. I was just curious if this ancient DNA can be used to somehow answer the question of climate adaptation, especially since these plants were the early plants were there during the previously happened major climatic changes. So it's just a bit of a general question.
Yeah, I think that's a really good perspective, and I don't think it's something that people have tried much just yet, but I think one issue with at least my data sets is that these are crops and that people are irrigating them if it's getting too dry. So the management by humans is a question, but I do think that using the same approach on non-crop species could potentially look at that, right? So if you were able to find indicator species or species that are really well understood, Arabidopsis maybe, find samples, maybe not archaeological samples, but somewhere in sedimentary records, so prehistoric ones. If you were able to find them and they're well preserved, it could be possible that you use it to try to answer some of those questions. I think it's just a matter of finding the right species and to have a good amount of knowledge about it and to know how it responds to climatic changes. But I do think that would be a really clever way to look at it.
Thank you.
Thank you. Any more questions? No? Okay, well, in that case, please join me in thanking Nathan for this wonderful seminar and discussion. Thank you.
Yes, and thank you all for your great questions. All right.
Okay.
Bye.